to all the attendees. I'm really grateful for you taking the time to join us here today. We are live in Madison, Wisconsin, and we're here to talk about really our NovaFold program that generates highly accurate protein structural models and its ease of use, talking about accessibility, and really exploring the data that is provided after a simulation. Before launching into NovaFold specifically on its workflows, I'd like to introduce the full lineup of our Nova applications in the Laser Gene Structural Biology suite. We have 3 applications currently in the Nova line. Our first is NovaFold, which we'll be talking about more today for protein structure prediction. More recently, we introduced NovaFold Antibody, which is a specific homology modeling workflow specifically targeting the modeling and generation of structural models of immunoglobulins. We also have NovaDoc, which through collaboration is an application utilized for protein-protein interactions and modeling the docking of those proteins. So here at DNASTAR, the question that we hear and really often ask when interrogating a protein is, where do we start? And specifically, where do we start when your protein of interest has no structural information available for you to utilize for modeling? Where to go for a starting template? And really, this is a critical question when working with a protein of unknown structure, as most protein biochemists and molecular biologists or geneticists often require access to an accurate structural model that can help guide any mutagenesis or engineering work to help resolve questions related to structure function. So now let's get started on our introduction to NovaFold. The purpose for developing this program here at DNASTAR and our work with the University of Michigan to commercialize the power behind NovaFold is the ITASer algorithm, which was developed at the University of Michigan in the Zhang lab. And so a few years ago, DNASTAR entered into an exclusive agreement with the University of Michigan and the Zhang group to bring to market their ITASer algorithm. And over that time, we've developed a very strong relationship with the Zhang group in order to deliver its highly accurate and automated program to researchers. Now, ITASer, as some of you may know, is a little bit different than most of the homology modeling applications that are on the market. It is unique in that it is a threading application that also takes advantage of some ab initio techniques. So this really offers researchers greater flexibility in that you're not reliant on using structural templates for modeling that have or require high sequence identity to your protein of interest. And over the past few years, we've developed flexible licensing options and also provide multiple points of access to the application while offering expert technical support. And really, this is very important in that, we can help guide researchers and where, the NovaFold application can help the most, and also, understand the results, that are out, the results from the NovaFold application. And so you can really have a great starting point for any future projects that you have going. So it's really kind of an easy answer to the question of why ITASer. I think this can certainly be best answered in looking at the results of really the last 5 CAST competitions, which is the critical assessment of structure prediction competition. And as you can see, I'm just highlighting the last 3 competitions where the Zhang group from the University of Michigan has been on top every time. This automated workflow of ITASer has routinely won the competition. It was really a no-brainer for DNASTAR to partner up with the University of Michigan to commercialize this application because us with the University of Michigan feel that you are you have the biggest advantage when starting with the best structural model for the design of your future experiments. And in fact, the Zong group has even had, almost equivalent success with a second algorithm called Quark, which has been the runner-up in these automated competitions. But the difference here is that Quark, unlike ITASer, does not use any templates at all for structure prediction. It's a little limited by the size of protein, but nonetheless, you can see it's still outperforming many of the groups that participate in the competition. So over the last year or so, we've released 2 new versions of our NovaFold application powered by ITASer. And these two releases offer flexible points of access to research that either take advantage of cloud computing resources or utilizing local resources on a Linux workstation. And here I've just kind of shown you kind of the interface that you would see when running accessing NovaFold Cloud through our Protean 3D application in LaserGene, or the simple commands that are required to run a prediction through a Linux workstation. And here is a brand new snapshot of our Nova Cloud application. So we've been developing and putting more and more resources on the cloud and providing more straightforward access. And here you can see this is giving researchers direct access to NovaFold through our web browser. And this can be accessed after just a very short registration. And so after running a prediction in these cloud applications, the result can be accessed either through Protean 3D by logging in, or can be downloaded via secure download from your client portal on our website. These days, the use of cloud applications is increasing more and more. And at DNASTAR, we take security very seriously. As highlighted in our white paper, DNASTAR Cloud Security, you can learn more about our services that are hosted by Amazon and the path your data takes throughout the process. And as you can kind of see here as we go through the steps, through your local computer, there is a login, there's an authentication of your credentials, there are data and instructions transferred to our cloud service, which is hosted by Amazon. Virtual computers are spun up to run your prediction. The calculations are run in total isolation. The result files are then encrypted for storage, and then you can certainly access very simply and quickly download the results of the predictions onto your local machine. So really it's all about taking advantage of resources that might not be available locally to you and not tying up any local resources for a structure prediction. So before getting into the visualization and kind of the detail of the results themselves, I just wanted to point out the differences in the flexibility offered by the cloud and local versions of NovaFold, because as you can see here, there are a certain number of optional parameters that you can set that are available in the cloud application, compared to the local Linux application. Kind of going through the parameters that you can select. Predicting ligand binding site and function. This is very powerful. This will use a process defined by the coach and cofactor applications within NovaFold to identify potential ligand binding sites, also identify the site residues that are considered to interact between the ligand itself. Also, based on structural homology to other proteins, make a prediction on the potential function of your protein of interest. You also have the ability to define the number of models that get generated. The default is 5. the number of threading templates that are used throughout the prediction. We have that number is typically set at 20. You have the ability to adjust simulation effort, which is kind of the longest part of the NovaFold process, to either go kind of very fast or to extend it to explore all options. Also, you have the ability to remove sequences at a particular sequence identity. So if you do not want something to be modeled on something with 30 or 40 or 50% sequence identity, you can have those removed if you want. Also, we have developed in-house an enhancement called Enhanced Template Search, where the template that is used to generate your models, the confirmation of that is explored kind of intensely to provide, in a lot of cases, a more accurate model than without using it. But within the local Linux workstation version, we offer researchers a little bit more control in their prediction. It is coming soon in the cloud application, but for right now, it's kind of focused on, I would say, the more advanced user of NovaFold, in such that researchers are able to define pairwise atom distance restraints between two atoms within the protein of interest. You do have the ability to add your own templates for the protein threading. You can also nominate from the non-redundant PDB library that we actually provide to you for the prediction, to make sure that before you run the prediction that template is kind of singled out. Also, you have the ability to remove any template from that PDB library prior to the prediction. So maybe you don't want any influence on your structural model from that template. So we've introduced kind of the many ways in which you can access NovaFold, given you a background on the development of NovaFold itself, and really the advantage that it has over homology modeling, in that you don't need a template with high sequence identity to your protein of interest. So next question after running the prediction is simply, now what? So I want to take a look at our viewing application in the LaserGene Structural Biology suite. It's called Protean 3D, which was developed to analyze protein structural models and apply various analysis methods to those sequences and to those structures. So here's a snapshot of Protean 3D and what you could expect to view when interrogating a protein of interest. As you can see, there's many there are options to adjust the view and apply the methods to a sequence, such as the one kind of viewed in the bottom right in the analysis view of antigenic regions or B-cell epitope prediction. So this application, right now, you're seeing a lot of the multi-window views, but it also has the ability to be a singular view, which you can tailor to your own preference. And this is the place to go to view your NovaFold results. So speaking of results, here is kind of the start of the overview of what to expect from the NovaFold tile that is generated after a structure prediction. In this report, you're provided a great deal of information about the prediction itself, the templates used throughout the modeling process, and provides really easy and straightforward access to features within the LaserGene suite of applications. So kind of going down through this list, what you're provided with after the prediction is run is that you get a set number of models. The default is 5, with not only the models themselves, but also with quality measurements, theoretical modeling score, and RMSD against the templates that are used. Also, you get details about the threaders that were used. Within NovaFold, 8 threaders are used, and we provide, as mentioned before, the non-redundant PDB library to be used as templates. Once the prediction is done, you probably want to take a look at structural alignments against the templates that were used. And it even provides you with a list of related proteins that you can do structural alignments with. And then also you can take advantage of the whole suite of applications within LaserGene in that you can do direct sequence alignments to related proteins or the templates that were used against your protein of interest, and that's highlighted in the lower right. Also, this is very powerful. You have, your delivered results about predicted enzyme function. And not only that, but also the predicted identity of any residues involved in involved in catalysis of the ligand. Also, you have direct access to gene ontology information through Amigo, where based on the structural folds, there are predictions made about the function of the protein. And to take it a step further, any other ligand information predicted by the cofactor workflow within NovaFold, as you can see in the bottom, you have a few different predicted ligands, confidence scores, template that was used, and then identifying the residues within your protein of interest that are thought to be involved in catalytic turnover. So let's get into kind of a real-world example. And this comes up a lot. It's, what if you're working on a protein that has no structural information and very little sequence identity compared to proteins that have structural data available? How do you go about generating a model? It can be a big challenge if you were limited to homology modeling. And that just kind of lies part of the power of NovaFold. So we ran a couple cases, couple protein sequences through NovaFold from B. microti, an aldolase, and a hexokinase. Kind of looking at the results in the report file, we see that if we look at coverage, we're getting decent coverage against the templates that were used to create the model, but the sequence ID for each case is fairly low. In one case, as low as 28%, and in the other, it kind of varies between 43 and 60%, depending on the template that was used. So that's pretty low and would certainly present a big challenge to any homology modeling application. But the result from NovaFold is that we were able to get pretty good models with solid TM scores and decent RMSD values when aligned to the top template. And this is really because NovaFold is focused on fold recognition and getting the folds correct and not concerning the predicted folds and not concerning it so much with sequence identity. And in both cases, NovaFold also predicted the sugar binding sites and in fact modeled in ligands to both of the models presented. So if you're asking yourself, okay, well, what is TM score and RMSD and how does it relate to what I can do with that model? And this is kind of a decent overview of giving you, a start to interpret the accuracy or the modeling score or the modeling score. As we can kind of see, as we get closer to a TM score of 1 and an RMSD of 0, we're getting a more and more accurate structural picture in that we can use then those models for drug design or screening. And as we kind of go down less accurate based on TM score and RMSD, probably we're getting into the range of now we can start looking at ligand docking. Certainly at modest accuracy, we're able to do mutagenesis design and active site prediction. And really the cutoff for TM score is about a 0.5. And that's where we're kind of confident that we've defined the domain boundaries of your protein of interest. We've gotten the topology correct. So now that we've gone through how to access the power behind NovaFold and the result themselves, I just wanted to give you a brief glimpse into the new Nova apps that were added to the LaserGene Structural Biology suite. The first is NovaFold Antibody. As I said before, it's the specific workflow designed for really the most popular set of proteins right now in terms of a lot of academic, government, and commercial research. And so we built an application to really model antibody sequences on existing frameworks, but doing de novo or ab initio modeling of kind of the key determinant of specificity of an antibody, the H3 loop. We provide the antibody frameworks. This is kind of a high-level overview of the process of NovaFold Antibody. And also what's nice is that in order to kind of guide the research to the key context of an antibody makes with an antigen, our application automatically annotates the CDR loops. Now this for right now is only available locally through Linux Workstation, but we're working to get that on the cloud as well. As with NovaDoc, which has been a very successful and productive partnership with the Bates Group at the Francis Crick Institute, we are taking advantage of the particle swarm optimization algorithm that the Bates Group developed that does not use a library of templates, but instead makes these docking predictions based on their swarm algorithm. It's been a top-ranking tool in the Capri competitions, as with ITASer and the CASP, and it really explores, the flexibility of each protein during docking. As with NovaFold Antibody, this is a locally available application only on Linux Workstation, but we're working to get it on the cloud as well. So I'd like to just quickly jump into a live demo of the access of our Nova Cloud applications, and specifically NovaFold. So let me just kind of, exit out of here. And as you can see, we go to our website. There's a nice, easy place where we host your accounts that we can log into. I'm just going to log into mine. And this is kind of the view that you would expect to see as a customer of DNASTAR. You have your account, the licenses that you own, genome templates and transcript annotation if you're doing any genomic work, but also we have the Nova Cloud services over here. Once you run predictions, your data is hosted right here securely. It also can be accessed through the Protein 3D application. But if you want to run a new prediction, very simple. I'm just going to grab a KRAS sequence. I'm going to cut it and paste into the sequence window, enter the prediction name, and if I want, I can set advanced parameters and enhance template search, predicting binding sites and protein function, the number of models that I want created, threading templates, utilize simulation effort, and remove any sequence templates at a certain percentage identity. Once I submit, we can see that my prediction is starting. It started here. I can log out from here, log back in within a day. Generally, predictions take 15 to 20 hours. The result will be available, and I'd be able to then view the result in our Protein 3D application. So I want to thank you all again for taking the time. If you have any questions about the application or licensing, feel free to contact DNASTAR. And Sharon, I turn it back over to you.
Hi there. Thank you, Tom. We have had some questions come in from our chat during the presentation that you just made. So I'll go ahead and ask these questions. There are 3 of them. The first one is, what about side chain modeling? Does NovaFold or do Protein 3D support that workflow?
Yeah, so, NovaFold is best used for single chain soluble globular proteins. And once those models are created in the Protean 3D application, at this time, you are not able to do any mutagenesis directly in Protean 3D, but you can certainly run mutant sequences through NovaFold for a straight-up prediction. But we are working and we're looking to our fall release to have support for sidechain engineering in Protean 3D, so you can do that directly.
Thank you. The next question that we have is, how does licensing work?
Right, so licensing for our local application or our cloud is a couple different options. Our most basic option is a term license. Also, we do kind of more per prediction licensing, and that's through our website. So if you're interested in running a prediction for academic and government researchers, the first one is on us. The second one is very inexpensive. So if you just have a couple of proteins that you'd like to generate models for, you can do that directly through our website.
Great. And the last question that we have time for today is, do you have a Linux version of Protean 3D? I know that you do have a Linux version of NovaFold.
Right. So, yeah, this comes up time and time again. Researchers want to take their NovaFold, Linux-generated prediction files and go directly into Protean 3D on the Linux workstation. The answer is not at this time. Right now, Protean 3D is a Mac or Windows application. So you run the NovaFold or NovaFold Antibody or NovaDoc applications on a Linux workstation, and the results can then be viewed on any Mac or Windows machine through Protean 3D.